So today I'll be talking about our discovery of pan-serotype dengue NS2B, NS3 protease inhibitors with in vivo efficacy. So as Alpha said, I'm part of the global health group at Novartis, and we focus on addressing unmet medical need for diseases that primarily affect low and middle income countries. We have multiple programs in targeting malaria, Chagas, sickle cell, and other diseases, but today's talk will focus on our work against viral threats. This work has been ongoing at Novartis since Zhu last worked at Novartis many years ago in Singapore, but was recently given a boost following the AViDD grant award in 2022. As Zhu mentioned, this set up the UTMB Novartis Alliance for Pandemic Preparedness, or UNAPP, which is addressing 3 different viral families, dengue and other flaviviruses, Henipavirus, and coronaviruses. And as Zhu said, we are trying to develop antivirals that could potentially be deployed during the next pandemic. However, for dengue, it's an immediate global threat given the rapid rise in cases over the past 2 decades. It's currently spreading into new countries, including the United States and other European countries. So with over 400 million, with potential for 400 million infections a year, this can cause serious issues with the healthcare systems in these countries. Luckily, about 75% of them are asymptomatic. However, that still leaves 25% or 100 million dealing with potentially flu-like symptoms and potentially even worse with severe dengue. So our hopes are to develop an antiviral that could potentially reduce the viremia to allow for a reduction in symptoms as well as the risk to severe disease. So Novartis has a robust pipeline of both novel and well-validated targets. As Zhu talked about earlier, we have our phase 2 clinical candidate, EYU-688, that is targeting NS4B. We also have polymerase inhibitors looking both at the nuke and the non-nuke pathway. And today I'll be talking about our efforts looking for a protease inhibitor. And this work started right when the AViDD grant award was distributed. So we've been working on this project for about 2.5 years. And because we have so many different mechanisms of actions, we are investigating the possibility of combination therapies in the future to potentially get broader serotype coverage as well as mitigate any drug resistance. So NS3 protease is an obvious target for dengue. Viral proteases are a very successful class for antiviral development with approved therapies in HCV, HIV, and COVID. It's responsible for cleaving the viral polyprotein and is essential for viral replication. Because it shares a high degree of similarity amongst the Flaviviridae family, we had hoped that we could get some broad spectrum activity against the other flaviviral family members. So NS2B, NS3 protease functions as a complex with the transmembrane protein NS2B. And you can see here in orange, this kind of wraps around the NS3 enzyme and is essential for correct folding as well as enzyme activity. In our initial analysis of the protein, we identified 3 potential binding pockets. So the active site has been extensively targeted by many, many groups, including within Novartis. These have typically resulted in the development of macrocyclic peptides because the pocket is very flat and featureless. However, these tended to have poor drug-like properties, and so I don't believe there's been any active site inhibitors that have been advanced to the clinic. We also identified 2 additional allosteric pockets. What we called pocket 1 sat at the interface between NS2B and NS3 towards the back of the protease here. And there was a literature compound that suggested that this could potentially disrupt enzyme activity. Pocket 2, however, sat closer to the active site underneath this NS2B beta hairpin. And a more recent paper suggested that targeting this pocket could potentially lead to in vivo efficacy. So while we were investigating approaches to target one or another of these pockets, we decided to go for a more broader screening approach to be able to target all 3 pockets in a single assay. So we enabled a high-throughput biochemical screen using a rhodamine-based fluorescent activity assay. This is a well-validated assay that is very robust and could be miniaturized down to 1536 well. And so we used an unlinked dengue 2 NS2B NS3 protease in order to allow for a more physiological range of conformation. And we screened the Novartis full deck, 1.8 million compounds at 10 micromolar, in addition to several smaller focused decks. We got about a 0.4% hit rate using a 50% inhibition cutoff, and we confirmed about 25% of these with an IC50 of less than 10 micromolar. Going into this, we knew that this rhodamine assay had the potential to have some false positives. So we enabled an orthogonal rapid-fire mass spec assay in order to try to reduce some of those hits. Here we took a natural substrate. It was a 9 amino acid sequence spanning the NS2B, NS3 cleavage site. And we were able to detect product formation using mass spectrometry. And when we looked at when we compared the 2 biochemical assays, what we noticed was that over 65% of our compounds actually show a loss of potency in the rapid fire mass spec assay. And many of these, about 95% of these compounds actually had a pKa of less than 6.5. So we were able to eliminate some of the non-specific acidic hits using this assay. In addition to the biochemical assay, we enabled 2 different binding screens in order to identify additional starting points. We ran a 2 billion compound DNA encoded library screen where compounds are linked to a DNA tag and then incubated with the NS2B NS3 complex. The bar-coded compounds that bound to the complex were then eluted off and then analyzed by next generation sequencing to identify the compounds of interest. Because they had the ability to screen a couple of different conditions in parallel, we were able to test dengue 2, yellow fever, and Zika in parallel in order to identify a broad spectrum inhibitor. Unfortunately, we were only able to see strong enrichments for the Zika complex here. We ended up resynthesizing 9 of these hits and saw actually strong biochemical activity with IC50s less than 100 nanomolar against Zika. This we confirmed binding by NMR and co-crystallized 2 of the compounds to the active site. However, none of these compounds had activity against dengue. And when we looked at a few of the hits from the dengue screen, those also did not have any dengue activity. And our priority was to get dengue activity. So we didn't initially follow up on these compounds. The other assay that we enabled was a native mass spec screen. This technique allows for the protein to remain in a native folded state and preserves non-covalent interactions, such as compound binding. Because this assay was medium throughput, we chose to run a 10,000 large fragment deck that was designed to have good solubility, phys-chem properties, and drug-like features. So basically compounds that a chemist might want to follow up on. The screen was performed by incubating 4 compounds per well, with each individual compound at 25 micromolar. And then the wells that were positive were deconvoluted, and the individual compounds were rescreened for binding. We identified 17 hits, including compound 1 here, which exhibited 36% binding. So once we ran all of our screens, we needed a way to validate and identify the location of binding. And we enabled a number of biophysical assays to do this, but our gold standard ended up being NMR. We were able to successfully characterize chemical shift perturbations for the active sites using compounds from the literature. However, identifying compounds that bound to the allosteric pockets proved a little bit more challenging due to the absence of a really good positive control. By mapping the amino acids, we were able to tentatively assign a few of our compounds to those allosteric sites. Unfortunately, they had poor ligand efficiency and drug-like properties, so we didn't follow up on those hits. Instead, the compound 1 that was identified in the native mass spec screen, we mapped that binding to the active site seen here. And then when we tested it against the Zika complex, we saw that compound 1 really stabilized this Zika complex. And this actually made it a good candidate for X-ray crystallography. And at the time, we were really struggling to get a crystal structure of any of our compounds with dengue. And so compound 1 actually became our first compound that we co-crystallized with an unlinked Zika NS2B, NS3. And we saw some novel contacts, some hydrogen bonds with tyrosine 161, glycine 153, and aspartate 129, as well as a pi-pi interaction with tyrosine 161. So this looked like a pretty good starting point. However, when we looked at the biochemical potency of this compound, it really didn't show great potency with about a 80 to 90 micromolar activity against dengue, and very poor activity against the other flaviviruses. However, our chemists noticed that there's this pocket here underneath compound 1 that was empty, and this is the S2 pocket. And our chemists believe that by extending this compound down into this pocket, we could gain some additional potency. And so, and that's exactly what they did. They were able to generate several compounds here that drove the dengue potency down all the way to 54 nanomolar against dengue. And you can see here compound 4 stretches all the way down towards NS2B. And this compound maintained the initial interactions of compound 1, but gained some additional interactions in the F2 pocket. And so now that we had some great biochemical potency, we wanted to be able to see if we could get cellular activity, which has been a longstanding problem in the field. And so we obtained the nanoluciferase flaviviral panel from Zhuping's lab at UTMB, where he inserted nanoluciferase into the capsid region. And this reporter viruses were very stable. So we established dengue 1 through 4, as well as Zika, JAV, and yellow fever. And by infecting HUH7 cells in a 384 well format and incubating for 2 hours, followed by compound addition. And 72 hours later, the luciferase was a readout for viral replication. And so here at Novartis, we have screened over 1,900 compounds against all 4 serotypes in the past 2 years, in addition to thousands more compounds screened as a counter screen for our other virology projects. So this has been a workhorse of an assay at the site. So when we put our NS3 protease inhibitor compounds in this assay, we saw a good potency against dengue 2. And we saw about a 10-fold biochemical to cellular shift which is about what we were hoping for. In many other cases in the literature, we saw kind of a reverse phenotype where the cellular potency was stronger than the biochemical potency. And this always led us to think that there were some potential off-target effects with those compounds. We also set up the Zika assay, and we did see potency against in the Zika cellular assay. However, there was a much bigger shift between the biochemical and the cellular assay. And we were never really able to understand why there was a larger shift with this assay compared to dengue. So now that we had cellular activity, we obviously wanted to see if we had in vivo efficacy. And I spoiled the conclusion to this with my title of the talk, but we took compound 4 and put it into mouse PK, where we saw a decent half-life, about 4 hours, good exposures, and good oral bioavailability. And so we took that compound and put it into a dengue 2 TSV01 infection of AG129 mice. And so this was a prophylactic treatment where we infected the mouse about an hour after the first treatment. And we treated for 3 days BID. And at the end, we measured viremia. And so you can see our compound 4 showed about a 3, 2, and 1 log reduction in viremia at 250, 150, and 75 mg per kg respectively. And so this was really kind of the first in vivo efficacy for a small molecule inhibitor against dengue that we could find in the literature. So we were really excited about this result, and we went full steam ahead into a lead optimization program for our compounds, where we were trying to address 2 big challenges. So one of our goals was to be able to target all 4 dengue serotypes. However, our weakest serotype was dengue 3. Our other challenge was permeability. You can see here comparing the dengue 2 cellular potency with dengue 3. Most of our compounds showed at least a 10-fold shift in activity, with some showing closer to a 100-fold shift. And we could never really explain why based on sequence and structural analysis. We were able to get a dengue 3 crystal structure, and there was nothing to really explain the difference in potency. However, our chemistry team was able to drive the potency of dengue 3 down past 100 nanomolar, but we still were struggling with permeability for a while as assessed by the MDCK assay. And only until the end of last year, towards the beginning of this year, where we started to see compounds in this green box here that had both dengue 3 potency along with good permeability. And so we continued to pursue lead optimization, and this resulted in over a 5-log improvement in potency with coverage across all 4 dengue serotypes. And this was primarily driven by taking compound 4 here, which I talked about earlier, and crossing it with some of the compounds that we identified in our high throughput biochemical screen. And this led us to compound 7 here, which showed a single-digit nanomolar potency against all 4 dengue serotypes. Unfortunately, we did pick up a little bit of a hERG signal, which our chemistry team is currently trying to address. We took these compounds and looked at PK and efficacy, and we saw that the enhanced potency and PK led to increased efficacy in vivo. So compound 4 here, we needed about a 75 mg per kg dose to get a 1 log reduction in viremia. However, as we progressed, increased the potency and increased some of the PK, we were able to drop that dose down to closer to 30 mg per kg to get a 1 log reduction. And compound 7 ended up needing less than 0.3 mg per kg to get a 1 log reduction. So compound 7, we're currently pushing down through our flowchart and into several safety studies, and we're continuing to move that compound forward, as well as optimizing some additional compounds to improve some of the properties. In parallel to the lead optimization efforts, we also decided to do some resistance selection pretty early on for compound 4. And this was done at UTMB by Zhenji and Zhuping's lab. So there hasn't been very many resistance selection done for this mechanism of action, NS3 protease, because there haven't been very, very potent compounds. And so we wanted to assess kind of what our barrier to resistance was early on. And so she took our compound 4 and did 8 replicates of the inhibitor-treated selections, along with 2 replicates of the DMSO control. And so she started at a pretty low EC50, 0.5x, took it out 25 passages and increased the compound concentration all the way to 10 micromolar, which is about 100-fold times the EC50. And she monitored the viral RNA by QRT-PCR in order to identify when it was appropriate to increase the compound concentration. And you can see here that the final passage 25 selections, the DMSO treatments showed minimal shift to compound 4, while the compound 4 selection showed about a 100-fold shift in EC50. And so she took all of these selections and did whole genome sequencing and identified mutations in both NS2B and NS3. You can see here in NS2B the presence of this serine 83, the mutation of serine 83 in all 8 compound selections and none in the DMSO selections, which would be a little concerning since they come up pretty early on. However, when we looked at the different serotypes, this threonine is actually present in the wild type sequences of the other serotypes that we tested. So we don't believe that this mutation would cause a reduction in potency because we're able to cover those other serotypes. In NS3, we did see some active site mutations appearing. So D129, tyrosine 161, and serine 131 appeared only at passage 25. So that's the highest compound concentration. And we didn't see those come up at any of the earlier passages. So with that data, we thought we might have a high barrier to resistance compound. There were some additional single mutations that were outside of the active site that appeared in a few selections. Those we think are potentially fitness-related mutation, but the assessments of the individual mutations is ongoing. So when we looked at the clinical prevalence of these mutations, here is just a straight dengue 1 through 4 serotype alignment, and here are the active site mutations boxed in black. You can see that the serine 83T mutation in NS2B is prevalent in about 58% of all the clinical sequences that we showed. But as I said, we don't believe this mutation will affect the potency of our compounds. The other active site mutations that in NS3 all show a very low frequency in the clinic, with only 7 sequences that were identified for serine 131 in almost 14,000 clinical sequences that were analyzed. The non-active site mutations also were at fairly low frequency, with the one exception of A120T. And this, similar to the S83T mutation, was also present in dengue 3 and dengue 4. So we believe we could probably cover this mutation with our inhibitor. So in summary, we took a screen anything that we could get our hands on approach, and we were able to identify diverse chemical matter with a novel interaction with tyrosine 161. We enabled a a whole host of biophysical and spectroscopic assays in order to validate our hits and to identify where they were binding. X-ray crystallography really enabled rapid improvements in potency, and our animal model was able to show one of the first in vivo validation dengue 2 mechanisms of action. The resistance studies performed at UTMB really showed that we are on target in cells, and we are currently planning for in vivo safety studies in Q3 of 2025. And I'd like to acknowledge the whole host of individuals that contributed to this project, without which none of this data could have been generated, both at Novartis and at UTMB. Genji, who did a heroic effort doing the resistance selection work at UTMB, as well as our chemistry team led by Joe Young, who was able to take an 80 micromolar hit and generate a compound with 1 nanomolar potency in the cellular assay in just about a year timeframe. And I'd like to thank the organizers for allowing us to share some of our data to the community. Thank you very much.